feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yonglin School of Medicine Healthy Longevity webinar. It is great to see you all in this show. Professor Michael Snyder is our main presenter today, and he will talk how to transform healthcare. And of course, everybody would like to do that. I'm sure that will trigger lots of questions, so please use the Q&A function. We are starting, as always, with a very short presentation, this time by Kalisha Shusri, our research assistant at the Center for Healthy Longevity and the Translational Research Program. And she will talk about a novel bioactive for liver, which might, maybe benefit aging and diseases. Over to you.
Thank you for the kind introduction. As mentioned, I'm Kalisha, a research assistant at the National University of Singapore's Center for Healthy Longevity. Today, I will present to you my research plans on trigonelline as a novel bioactive for liver benefits in aging and disease. The hallmarks of aging are some of the occurrences that drive the aging of humans. By understanding these hallmarks, we can develop therapeutics to promote healthy longevity and combat age-associated diseases. One molecule linked to these aging hallmarks and aging disease is NAD. In fact, trials involving small cohorts and preclinical work have shown that NAD levels in the muscle and brain decline during aging. Hence, many NAD precursors have been utilized to boost NAD levels. As shown in this cartoon, you may have heard popular supplements such as nicotinamide riboside or nicotinamide mononucleotide, which increase NAD levels through the salvage pathway. However, work by my PI, Vincenzo Sorrentino, has identified a new precursor that increases NAD levels through the Price-Handler pathway, shown here in the red square, which normally utilizes niacin or nicotinic acid in the context of human disease. The study, which was conducted between Nestlé Research and NUS, discovered a depletion of a molecule called trigonelline in serum samples from Singaporean patients afflicted with age-related loss of muscle mass, or sarcopenia. The levels of trigonelline correlate with NAD levels in muscle and link with muscle health. Grip strength increases along with trigonelline levels, suggesting potential health benefits for muscle longevity. So what is trigonelline? Trigonelline is a natural alkaloid found in humans, but largely produced in plants such as coffee beans by methylation of niacin. So if you drink a lot of coffee, your blood will have higher levels of trigonelline. It may be converted into NAD, although the mechanism has not yet been elucidated yet. However, we do know that this conversion requires the enzyme NAPRT involved in the Price-Handler pathway as for niacin. Indeed, trigonelline administration in mice increases NAD levels in multiple tissues such as the liver and muscle. NAPRT is also highly expressed in the liver compared to other organs, and given the central role of the liver in NAD metabolism, it suggests that in fact trigonelline may also exert benefits in the liver. Thus, the focus of my research project at NUS will be on utilization of trigonelline as a novel NAD precursor for benefits in aging and disease. In Singapore, liver aging and disease are mostly linked with non-alcoholic fatty liver disease and non-alcoholic steatohepatitis, which indeed affects 30% of Singaporeans with increased prevalence in elderly people. I will be investigating if trigonelline administration is able to improve liver health during aging and ameliorate these diseases. To do this, I utilize multiple in vitro and in vivo models, allowing me to conduct mechanistic investigations as well as physiologically relevant effects of the bioactive on the whole organism. For animal experiments, I'll be conducting a chronic study in aged mice to observe the effects of trigonelline on liver health during aging. I'll also be using the nematode model to investigate conserved pathways of lipid metabolism that are present in the mammalian liver too. I'll be looking at 2D liver cell lines as well as primary hepatocytes from patients, as well as the 3D organ-on-a-chip platform to model NASH. Here is a very early result I have obtained from the liver-on-a-chip experiment, made possible with the kind support from the Henry U Lab. Shown here is a liver organoid developed from human-derived stem cells. The organoid is stained with body P, which stains for lipid droplets, and we induce NASH in the organoids using a cocktail of fatty acids and fructose. As you can see, the NASH organoid has high body P staining of lipid droplets. However, when we treat the organoids with trigonelline, the intensity of the lipid droplets decreases, suggesting lower fat content of the liver organoids. Such preliminary results are promising, but I have many more experiments currently in progress to confirm this in the course of my research plans. Thank you for your attention, and I welcome any inquiries through email. We are a new and growing lab here at NUS and just starting out our experiments, so I look forward to presenting to you again soon.
Thank you, Kalisha, for this very nice results. I think we would really like to know how we can boost our NAD levels, which are associated also with clinical outcomes. Now it's my great honor to present and to introduce Professor Michael Snyder. And he is a real pioneer in precision medicine. He is the chair of the Department of Genetics and director of the Center for Genomics and Personalized Medicine at the Stanford School of Medicine. And Professor Snyder received his PhD at the California Institute of Technology and continued then as a postdoc at Stanford University. And I can tell you, he is really the leader in the field of functional genomics and proteomics and one of the major participants of the ENCODE project. He has published an enormous amount of papers, over 850, and is one of the most cited scientists worldwide in this field. Professor Snyder has co-founded 17 companies with combined enterprise value of over $6 billion. So it's an honor to have you here, Michael. We are really looking forward to your talk, talking about transforming how we can transform the healthcare using deep data and remote monitoring.
Great. Well, thanks very much for having me here. It really is a pleasure to show you what we're up to. And as you just heard, it really is our mission is to use big data to transform the healthcare system. So we believe the healthcare system is broken. First of all, we tend to practice sick care rather than truly healthcare. But more importantly, even when we practice healthcare, if you think about the steps we do are very antiquated. You typically will get in a car or bus or something to travel to a doctor's office, almost always at an inconvenient time. You'll arrive at a physician's office that looks pretty much the same as it's done for the last 40 years, maybe with a few new gadgets. When you're there, they'll put a needle in you that usually hurts, and they'll draw a very large aliquot of blood. And from that blood and the time you're in the physician's office, they actually don't make very many measurements when you get right down to it. And then from those measurements, they make decisions about your health based on population averages. And we think every single one of these steps can be improved. And just to give you an example of this last one, treating people based on population averages, you've probably been told since you're little, well, at least if you were in the US system, British system, that your temperature when you get ill is 90, or your temperature when you're healthy is 90.6. And it turns out, first of all, that number is wrong. The typical number is more like 97.5. And this is one such study. There are many very similar ones. But the more important point here is there's a spread. This is a 25th quartile, 94.6. This is a 75th quartile, 99.1. And what that means in today's world, if your normal baseline temperature is 94.6, and you go to a physician's office and they measure 98.6, they're going to tell you you're healthy. Everything's normal. You're fine. Go home. But the reality is I'm quite confident if you're 4 degrees over your baseline, you're ill. And so that's a big theme of our work. Take measurements and know what your baseline is so you can see shifts from that. And you'll see in a number of cases how that's proven to be useful. So your health, you probably know it's influenced by many things. It's influenced by your DNA, which we can now determine, but all these other things influence your health as well. Activity, food, stress, exposures. We know intuitively they impact your health, but we're in a world where you can now measure these sometimes pretty well, like activity and things like that. We'll talk about that. Some things are still a bit clunky, like food and stress. But even if you don't measure these directly, you can measure their impact on your health by doing deep data dives. And that's thanks to a revolution in genome sequencing and mass spectrometry for being able to measure lots of molecules in your blood and your microbiome and such, and even wearables. And so we'll talk about all that. So using these advanced technologies, we actually a number of years ago set up something we call personal omics profiling, where the idea is that in addition to sequencing people's DNA to predict disease risk, we would actually collect lots of data out of their blood and urine. These are typically out of your immune cells in the blood, your epigenome, which your DNA methylome, your transcriptome, proteome. And then out of plasma, we'll also measure proteome, cytokines. These are important immune molecules, metabolites, lipids. We'll do deep data measurements, again, out of your blood. These will also measure out of your urine. We'll also follow your microbiome, which you may know is very important for digesting your food and making essential vitamins, at least the gut one is. But these days we follow 4 different microbiomes. We're collecting samples for urine and haven't analyzed that yet. And that's turned out to be quite interesting. So we do these deep, what are called omics measurements on top of pretty deep clinical tests, questionnaires, some advanced tests like stress echocardiograms, glucose control measurements, and you'll see why in a minute. And then we also, a number of years ago, 10, I guess now, we basically added on wearables and biosensors. So we're taking, one aspect is we're taking very, very deep data on people, and we're also doing it over time. We'll actually sample people every 3 months while they're healthy. And then if an adverse event comes along, like a viral infection, what have we typically take 5 to 7 additional samplings. And so we've been following the smallest cohort of people, 109 people, for actually over 10 years now. I'm one of them. I've been profiling me for 13 years. And you might say, why are you doing all this? Well, we're trying to understand what it means to be healthy, not what it means to be sick. What does it mean to be healthy? How does that change over time? How does that differ between different people? How are all of us on this call different from one another? What happens at the earliest times of illnesses like a viral infection? What does that look like? And then perhaps most importantly, from a health perspective, can we use these advanced technologies like genome sequencing and deep data dives on people's blood with mass spec? Can we use these to better manage people's health? And with regard, I can tell you, it's controversial when we started. People really didn't like us sequencing genomes of healthy people. We thought we were going to turn everybody into hypochondriacs. But anyway, we went ahead and did this. And I can tell you just from the first 3.5 years of profiling the small group of 109 people, we had 49 major health discoveries. So nearly half the people learned something pretty important. And it was spread over a wide range of areas: heme-oncology, cardiovascular disease, metabolic disease. And many of these turned out to be a pretty big deal, meaning we caught some with early lymphoma, 2 people with precancers that can convert to aggressive cancers. They get screened more. 2 people with serious heart issues, one picked up by genome sequencing, one by wearables. And all these were found asymptomatically. Okay, so before people had symptoms, we could see something was off. And not only that, it wasn't any one technology that found these things. Sometimes it was genome sequencing, as in the case here where we found this heart problem. We saw the mutation and follow-up tests that this person actually had a heart defect. His father died actually of a heart attack. And then in other cases, a wearable. Some cases it was imaging. Often it's a combination of markers like imaging plus some molecular markers. And so the way we like to think about this, if your health is a 1,000-piece jigsaw puzzle, we're trying to take 500 or 600 pieces so we can get a much clearer picture versus 5 or 6 like they do today. It's just woefully inadequate in our opinion. And so these deep data dives, again, found many, many things. The other aspect is, and I can dig into more details, and happy to do so if you have questions, genome sequencing, all these technologies were quite powerful. But the other thing, because we were profiling people over time, we can actually see how they were aging. And it turns out everybody ages differently. Like this person, their top pathway that changed was the cardiac hypertrophic signaling pathway. They were a cardioager, and we later learned this person is stage 2 hypertensive. So sort of fits with that idea. This person over here, they actually are a typical ager. Their coagulation, metabolic, and other pathways gradually go over time. This is 4 years of data. We think you only need as little as 5 measurements within 2 years, and we can tell you how you're aging. And so in the end, we classified people into agetypes. We were statistically empowered to be able to find 4 of those. I know there are more than 4, like the cardioager, but we only have 1 of those. But anyway, we like kidney, liver, metabolic, and immune. These are all agetypes, if you will. And so every column here is a different person. Every row is their agetype. And so what you'll notice over here, this person in this box, they're a kidney and metabolic ager. This person's a kidney ager, and so on and so forth. You get all the way to the right, folks over there are actually aging in all 4 categories. So we can see how people are aging. It turns out there are clinical markers associated with these agetypes. So we think the information is actionable, and you could potentially modify that. And we've seen people do that in our study. So one question I get asked a lot is, well, this is a research study. It is. We're doing these deep data dives. Is it actually, how are you going to scale this? It costs us a lot of money to do this. Well, I'm in Silicon Valley, so the way you still scale things is form a company, and that's what we did. We formed this company, QBio, that does a medical version of what I showed you. It's deep data, much more actionable information than everything, just measuring everything. It also does whole body MRI, and we think that's a big deal. If you talk to any physician in the United States today, ask them, should you do a whole body MRI? They'll tell you no, because you'll have nodules. Women will have them in their ovaries, men in their prostate. And I would say, yes, the company would say, yes, you should do this, because the issue isn't do you have nodules. Everybody has nodules. The issue is do you have any growing nodules? And the way you know that is by these longitudinal measurements. So that's a big, important part of our work. Profiling people over time lets you see shifts. And that's turned out to be true in those 49 discoveries I told you about. Some were picked up because of longitudinal shifts. And that's true here in QBio. These are just some of the first cases. These were also all found pre-symptomatically. Early ovarian cancer, prostate. We even found some with early pancreatic cancer, which is almost never found earlier because of this longitudinal profiling and so on and so forth. So again, many of these we like to think were life-saving by having found these. And again, all found pre-symptomatically. So one of the other things we've been working very hard on is this idea, all right, well, you go to a doctor's office to get measured. And if you think about it, why do you have to do that? For many things, you shouldn't have to do that. Certain things you will. You're not going to go do a whole body MRI at home. But there are many measurements you could make at home. And so we've been working on what we call Amazonizing healthcare, the idea of trying to do this at home, much like you would do your shopping. So we've really been pushing 2 areas. One is wearables, and the other is this microsampling field. So wearables, came on about 10 years ago. When they came out as fitness trackers, we recognized, well, these are actually pretty powerful health monitors. They're mostly smartwatches, but there's all kinds of devices out there, smartwatches and rings, I'd say these days. They're powerful because they're measuring you 24/7, and they measure lots of important things. Resting heart rate variability turns out to be important, skin temperature. Different devices measure different things and at different accuracies. I'll argue even if they're not accurate, they still work pretty well because they'll see these shifts, and that's what really counts. So we actually got involved with us back when they were first just fitness trackers. We said, well, these are powerful health monitors. Let's put them on our group of people and see what we can learn. And right away, we discovered, actually, in my case, I found my Lyme disease, of all things, with a smartwatch and a simple device called a pulse ox that measures your blood oxygen. And the story behind this was helping my brother put up fences in rural Massachusetts, and then 2 weeks later was flying to Norway. And on that last flight, I saw my blood oxygen drop. It normally drops on airplane, but it dropped way low, and it never came back to normal when I landed. And I also saw my this is low resolution, but I saw my heart rate was up. I later learned my skin temperature was elevated as well. So I found all these, and these are just with simple smartwatch and a pulse ox. And so this, again, it was all found pre-symptomatic. I spotted these things going on. Then I got symptoms off and on. I finally went to a doctor in Norway. He drew blood, saw my immune cells, monocytes were up, said, yep, you got a bacterial infection. You should take penicillin. I said, no, I should take doxycycline. Got a bit tense there, as you might imagine, because no physician likes to be told by their patient what they should be doing. Anyway, he did give in. I took it basically here, cleared it up right away. You take it 2 weeks. And when I got back, I was tested. Sure enough, I was Lyme positive. And I just happened to have given blood before I left. It's not random. I measure myself a lot. In fact, here are my fours. Well, one of my watches just broke. So I only have 3 smart watches and a ring right now. So you can tell I'm a big believer. I also have a glucose monitor. We'll talk about those in a minute. Even my hearing aids do all kinds of cool measurements. So the point, in addition to hearing, so the point out of all these is that we could tell when I first got this infectious disease with a simple, wearable device. And then we went on, I won't show you the data, but we could show that we could tell when people were getting respiratory viral infections. In my case, I had 3, one of which was asymptomatic. And then we had other people that had these as well. And in every single case, we wrote algorithms. We could see a shift up in resting heart rate prior to symptom onset with a simple smartwatch. It's very powerful. It doesn't work as well for skin temperature because not everyone keeps their device tight, but it works very well for resting heart rate. So we published this in 2017. We're excited about it. We're building out, improving our algorithms, building out a database to do this at scale. And of course, along comes COVID. And so if you think about the way we try to detect COVID and other things these days, are the first line, if you will, is usually to use a thermometer, which is a 300-year-old concept. And, we do have better, newer devices. I wouldn't say they're better with these UV ones, but they're not, they have their limitations. Speaking for myself, they usually shine it on me when I just came in from the outside and I measure 33 degrees centigrade, which is ridiculous. They weighed me in anyway, even though it's a worthless measurement. So this is just not the best way to do things. And PCR and antigen tests, they're great, but you don't do them all the time because it's just not convenient or practical. So the bottom line is we think smartwatches resting heart rate could be a powerful measure. So when COVID hit, we quickly partnered with Fitbit, enrolled 5,300 people, 5,300 people. And we basically found 32 that had they were wearing a Fitbit and they got COVID and they had both a positive diagnosis date and a symptom date. And this was our very, very first case. This is just showing resting heart rate. And basically what you're supposed to notice, this is a diagnosis day here, day 48, symptom day 47. But their resting heart rate jumped up 9.5 days prior to symptom onset. You can't miss that signal. Here's our real-time detection I'll tell you about in a minute. So the point is that we could see this jumping up in advance. And overall, it worked 80% of the time, 26 of 32 people. We could see their jump up in resting heart rate in advance of symptom onset. And the median was 4 days prior to symptom onset. COVID has a long pre-symptomatic period. So it's actually a good disease to pick up with a smartwatch. By the way, you can enroll in our study, I believe, even in Singapore or in Taiwan, actually anywhere in the world, I believe. So the point is that you can pick this up by enrolling in the study. So again, it worked 80% of the time. So then we built this real-time detection system. What it does is it follows your baseline on a circadian fashion and looks for a jump up in resting heart rate and some other signals, sleep. We're going to try and bring in more data types now, but it brings in a ton of data and it's analyzing in real time, at a personal level. It's an anomaly detection system. It looks for a shift up in resting heart rate over your background. And it works. This is one of our very first cases. Same number works 80% of the time. Here's an individual, their symptoms appeared here. They were diagnosed officially the next day, but they were getting red alerts, if you will, alarms, 3 days prior to symptom onset, again, in advance of symptoms. And so it works for Fitbit, works for Apple. I think it'll work for all devices, just that we have most of our examples with those. It works pre-symptomatically, as I say, 80% of the time. It also works even in asymptomatic case. There's an example here with Fitbit. Same is true for Apple. This individual was diagnosed with COVID here, but they were getting red alerts for basically 2 weeks prior to symptom onset. Prior to diagnosis, and never had symptoms. So again, it's a very, very sensitive measure. You only need as little as 2 beats per minute. We can pick it up with a smartwatch. Now, it's not specific for COVID because other things will trigger this as well. It's basically a stress detector. In workplace, stress is the number one trigger of red alerts. Okay, so psychological stress is probably the toughest. Other things like intense exercise, travel, these will trigger, but these you can kind of figure out contextually, you know. So if you run a marathon, your heart rate will be up several days. Resting heart rate's the number one trigger of the signal. Even other infections, here's mycoplasma we can pick up, all with basically a simple smartwatch. So I can tell you it worked on me personally. And one of the things that we've found in my case, and actually several others, is that it's more sensitive than an antigen test. And this is my case. So I mostly have green days, but I had a particular family stress event with some travel for, 2 red alerts. And I woke up one morning, I was getting ready to go to New York City. I was congested. And I'm thinking, is it just a little bit? It's April. I'm wondering if it's allergies. What's going on? So I do an antigen test and I'm negative. That's right there. I look at my smartwatch and I'm positive. So what I do, I went on the plane anyway to go to this meeting. And the next day, in order to go to the meeting, you actually have to get tested again. And once you know it, I'm bright positive. So I wasn't even able to go to the meeting. I spent a whole week in New York City in a tiny little room, all because I listened to my antigen test and not my smartwatch. So smartwatch is more sensitive. It's less specific, but it can tell when these things are happening. So we think this is just the tip of the iceberg. You can tell a lot of things with a smartwatch. You can tell your red blood cell levels. We use machine learning and other approaches to be able to see what kinds of clinical signal measurements we can get from a smartwatch. They're not clinical grade measurements, but they're good enough to see a shift. So you can see when you're shifting red blood cell, hemoglobin levels, even hemoglobin A1C and fasting glucose gives a signal with a simple smartwatch. And there's a lot of reasons why it does and makes sense it does. So again, we think these are going to be powerful health monitors for many, many things. This is relevant for diabetes. And the red blood cells very relevant for anemia. So other sensors, of course, many of you may know that are super powerful are these continuous glucose monitors, getting even more monitors of this type coming out. So these had been used for type 1 diabetics and insulin-dependent type 2 diabetics. And our claim to fame is we started putting these on normal people and pre-diabetics. And once you know it, there are some normal people are normal, meaning they have very good glucose control. They don't spike much in their levels. But then there are people who are moderate spikers. So their glucose goes up after eating. And then there's severe spikers. Some people classified as normal are just as bad spikers as type 2 diabetics. So they're really dysregulating their glucose. And so these monitors, which measure your glucose every 5 minutes, are super, super powerful. So we went on then to write algorithms, classify people into glucotypes. So we classified people into low, medium, or high glucose, if you will, spiking and measurement levels. And then we went on to actually show that different people spike to different foods, as did Aaron Siegel. So we both had found this, and this is something from Aaron's work. So it turns out that some people will spike to bananas, others pasta, some to potatoes, some to bread, some to white bread, some to brown bread. We're all very, very different. And your microbiome, Aaron had shown, is at least in part responsible for this. And so this is a good example. So here's an individual who spikes a banana, but doesn't spike to a cookie. Probably the only person on the planet that doesn't spike to a cookie. Most everybody does. But this person actually spikes a cookie, but not a banana, and that's much more typical. So again, people spike to different foods, and you can measure that with this glucose monitor. We had found that some people will spike to bread and peanut butter, others are protein bars, which have carbs in them. You should look at what's on those labels. And we also found that 80% of people spike to cornflakes and milk. Whatever you do, don't eat that stuff. It's like poison, not good for you. Probably should be banned. Could even be worse than smoking. So watch out for this stuff if you don't remember anything else from my talk. So again, these are very, very powerful measurements. So the other thing we've been working on is this idea about trying to do biochemical sampling at home and then collect samples at home and then get back deep measurements. And so we set up a remote monitoring system with microsampling. We spent about 6 years working on this. I know what it's going to sound like, but ours does work. The idea is that what you do is you prick your blood with a simple lancet, and then you collect very fixed volumes. And it turns out these devices, another one called TASA, are very, very powerful for this. And so what you'll do is you collect these samples, you send them by overnight to our lab, and we can measure all your lipids, metabolites, proteins, and even targeted assays we can run on these two simple little drops, 20 microliters of blood. And we ultimately measure over 2,000, about 2,200 different molecules in your blood. And so we went on to show that these are actually, most molecules are stable, certainly the proteins and metabolites. Some of the lipids go off, and we know which ones are good and which ones aren't. So then we went on to run two really, really fun studies, and they're shown here. This was the first one. We actually had 32 people drink this Ensure shake, a very simple mixture. And we sampled them before they drank the shake at 30 minutes, 60 minutes, 120, 240 minutes after drinking the shake. And not surprisingly, hundreds of molecules change, over 1,000 actually. And we can follow that. And many of these are pretty important. These are actually glucose control hormones, C-peptides, which is derived from insulin itself, GIP and incretin, and this other one. We can quantify this in people, and we can also see even inflammatory markers like interferon and such. So one of the things that we discovered is that everybody reacts to this same shake very, very differently. So they're all drinking the same shake, but they respond differently. And so that's shown here. Each box is a different person. I'm just showing you 3 carbohydrates. They tend to behave as a class. So 3 different carbohydrates. This person, it's a time course here. Their carbs plummet, go way, way down after drinking the shake. This person's go way, way up, way up, up. This person's kind of the same. This person goes down. This person goes down. So again, everybody's reacting differently. And we can group people into 5 kinds of people, 5 different groups. And so in gray here is the average, and these are the classes of molecules. So this group of people, it turns out, again, some of them won't spike much of carbs, others do. This group of people, their inflammatory markers go down when they drink the shake. That's shown here in this dark brown circle. But for this group of people, their inflammatory markers go up, it elevates. Same with this group, goes up. So for some people, the shake is pro-inflammatory, and for others, it's anti-inflammatory, suppressing inflammation. Again, so we're all reacting differently. We're trying to figure out what the basis of this is now. The other thing we've been able to do with this is actually do what I like to think is the ultimate experiment. We had one individual who gave 98 samples, basically one every waking hour for over 7 days. So 98 samples in total. They did this while wearing a CGM, while wearing a smartwatch to measure heart rate and other sleep and other things, food logging, and they did the microsampling, as I say, every hour. And then what we did was we looked for associations between molecules with other molecules and with the wearable data like CGM and other things. And we do a time-lag correlation because we're trying to see what kinds of events precede other events. The early events are more likely to be causative for downstream ones. Much like, if you walk, your heart rate goes up with the time delay. So what we found is that there are, not surprisingly, thousands of associations between your molecules and even some simple things like heart rate, steps, and glucose levels. And so, there are just many, many kinds, and they're very interesting. It's a very rich data set. And what we discovered is that, like in this individual, you can quantify some known things like the insulin C-peptide goes up after your glucose goes up. And it turns out in this individual, it's exactly 10 minutes is when that peaks. But other things related too, we found a new inflammatory marker goes up in response to glucose. This one's quite interesting. This is a protein involved in Parkinson's and dementia, alpha-synuclein. And it turns out that does correlate with glucose, but it turns out glucose goes up after. Well, you can see there's a bit of a pattern here. We actually think it correlates with stress, that a certain stresses will trigger the alpha-synuclein. And so we think if you're at risk for dementia or Parkinson's, that might be useful to know so you can modulate your activities there and better control your synuclein levels and maybe push off having dementia. Don't know. That was the hypothesis. So what do we do with all the data? Well, we've written a dashboard to pull in all your data, your wearable, your microsampling, your clinical data, can all pull in the single dashboard that you can visualize at whatever resolution you want, monthly, daily, weekly, what have you. And that's where the alarming system comes from as well. And so we think this is the future of health, that we will be in a world where we're actually going to sequence people before they're born, make predictions of blood, do deep biochemical and wearable measurements, and then better track people's health at a personal level for predicting risk, diagnose disease early, monitor, treat, all based on big data approaches. And again, one thing we do is we actually, to get this out to the world, we spin off companies. You heard already, I founded 17 companies. Everything I told you is highly conflicted. But here's a sequencing company. All the ones in boxes are related to this work. So this is our way of translating and trying to scale it. This is a continuous glucose monitoring company. By paraphrase, is the big data one I mentioned. These are some of the microsampling rhythm for treating long COVID. Turned out to be quite interesting in yellow as well. So that we think these are all going to be quite powerful. And here are the people who did the work. The wearables team, when it was at its peak, was led by quite a few people: Xiao Li, Amir, Tejas, and others. And the microsampling was led by Xiao Tao Chen, Ryan Kellogg, along with others as well. And I'm happy to take questions.
Thank you so much, Mike, for this fantastic talk. It's really music to my ears that you're focusing on clinical, biological, and digital phenotyping. That's what we really try to implement hard in longevity medicine, really optimizing health of individuals. So music to my ears. But having a longevity clinic myself and implementing that and dealing with technological difficulties, I would like to get your opinion. How do we transition the physicians into the new area of healthcare?
Boy, great question. They're they're one of the bottlenecks right now, because they always react terribly to our work. When we start with wearables or instant reactions, oh my gosh, those things are so inaccurate. And they said, well, actually, they're more accurate than what you'll measure in a physician's office. So we are, and we showed that in a paper, that if you pull someone's resting heart rate off first thing in the morning, it's way more accurate than what you'll measure in a physician's office. So I think we do have to educate physicians. I think that's part of it. To be honest, we're probably going to have to grow a new set, because it's hard to educate those who are, you're excluded, of course, Andrea, from this, but many people aren't so enlightened that there's a tendency to push back. And so I think we have to have people receptive to this. We have to show they work. Everything has to be evidence-based. And of course, the biggest bottleneck these days is cost. Who's going to pay for this stuff? Nobody pays, at least in the US, nobody pays for you to keep your people healthy. They tend to pay, like as the head of the hospital. So Mike, I only get paid when people walk in the door, and they're not going to walk in the door unless they're sick. So we need to shift that mindset. We need to get physicians involved in that same kind of thinking, not just say, oh, your heart rate went up again. Because on a wearable, you can ignore that. That's actually not necessarily the right thing. We need the right algorithm. We need to make it easy for the physicians. I think that's another factor. Because as you know, physicians have a total 15 minutes, at least in the US, to spend with their patients. It's not nearly enough time. And so I think physicians are going to have to see summaries of data to get brought in. And I think, to be honest, I think citizens themselves are going to have to take responsibility for their own health. I think that is the future.
Yeah, so there was lots in there already, but let me go back to the costs. I was quite impressed that you could predict the onset of COVID and in yourself, the Lyme disease. I did it, by the way, for myself with dengue. I already knew two days beforehand that yes, would have the spike. So maybe we can implement that in Singapore here.
I want your data. I want your data to look at that, compare it with the data we have. Sorry to cut you off. Keep going.
No. But especially the COVID papers you wrote, have you analyzed how much cost effectiveness that would bring, because not spreading COVID through just contamination because you are out there, and being in isolation much earlier? Did you try to do that?
We did a modeling study on this. It's a lot trickier than you think because of all the other red alerts and things that happen. So you have to build certain assumptions in. Like most red alerts, you can, like you say, you can contextualize and ignore. So how do you tell the ones that are from mental health stress versus, say, respiratory viral thing like COVID? that you can't right now with our system, but I know we can in the future. I think when we bring in respiration rates, things like that, we're going to be able to distinguish it. So I would view it in part that we're still pretty clunky at it, but I think, you hate to put time estimates, but it'd be awfully surprising in 5 years we can't get this working really quite robustly, that at least give people a heads up, like yourself with your dengue. When you see this thing jump up, not to, well, dengue is probably not so bad, but for COVID, respiratory viral things, you really do want people to not go jump on an airplane. So I think we can take, and then, so we haven't modeled it from the spreading fashion. We have modeled it from sort of the FOST pause rate, what it costs us, and what would it take to follow spread at a national level if right now in the US, 20% of people wear smartwatches. So you can actually follow spread. You can model this out, how quickly you could detect it. And it does work. And it is, it does cost some money. But I think there are ways of reducing money. This gets some technical issues. I guess I'll leave it at that. But so I think the way to look at it right now, just pretty early to be able to do this stuff. In the future, we should be able to get it cheap and everywhere. And I think it'll just be like a dashboard on your car. You pull data in, it follows you passively, and then alerts you when things aren't quite right.
Yeah, absolutely. So going a little bit from the predictive power of biomarkers, you already mentioned that dashboard. I love it. Going a little bit to another topic, and that's optimizing health, because that's what we try to do in healthy longevity medicine. So we don't accept that you are just normal, but how can you make, because of based on your makeup, the genome, the epigenome, etc., how can you optimize your function and fine-tune metabolism? Can you respond a little bit? How would I know that I am optimal, what the capacity of my body is?
That's a great question. I think, fantastic question. This is really, I think, a major hole in the longevity field. We don't really measure people well for this sort of thing. So the best markers are the methylation markers, in my opinion. C4VAS, clocks and things like that. I think they're decent. They're a little bit slow, and they're not as actionable, say, as the age type information we're getting. So I think shifting this over to metabolic tests will be very, very powerful for that. But I think backing up a little bit, I think our vision on this is that we're trying to extend people's health span. So the goal is have people live healthy lives and then, just pop off. So that would be the optimal way. And I think by monitoring your health, you can track, how things are going. You certainly see these shifts. Like I mentioned, my Lyme disease, my type 2 diabetes, I didn't talk about, is predicted from my genome sequence. So I figured it out. I saw it happen right away. It came up after a nasty viral infection, which, by the way, is now showing up with COVID. You may know 2 to 4% of people who get COVID wind up type 1 or type 2 diabetic. I'm the first case for type 2 to be shown. And it was predicted from my genome. So I think you can predict risk, you can catch disease early, and then that helps mitigate things. If you let disease get too far along, as I'm sure you appreciate, it's hard to reverse. So the lymphoma case we caught early, that person's in, complete remission, never had an issue since, found quite some time ago. Precancers get screened more. So I think the way we're doing now, we will measure people and find shifts, but I also think in the future in a sense, we have what we call a healthy signature. That's where these deep metabolic tests show up. They kind of tell you're green in all these different categories. We do it now in a physician's office, but the measurements are just so incomplete that I think we miss a lot of stuff, right? The 15 markers will measure in doctor. It's just not enough.
Yeah. And most of the times in conventional medicine, we are very happy if our numbers coming out of the blood screens are not bold and they are not red, because that means that's normal. But how can I make sure that I'm in the normal space, but I am in the optimal space? So did you do studies where you link it back to the genetic of that individual or other health signatures to then define for that individual that is optimal and you would like to reach that?
Yeah, boy, that's a great question. I guess to some level, we are trying to tune things based on known parameters. We want people to be in the good range for their cholesterol and all these various markers that are associated with health. But I think everybody's baseline, as you point out, is very, very different. And obviously, we want people to maximize that. My own belief is that the number one marker for longevity, you may feel the same, is VO2 max. It's the best predictor of people's health span. Of course, you don't do it all the time. It's not terribly convenient, but it is a good measure. And so if we can try to get people to optimize their VO2 max, and that does require experimenting. I think what nutrition is best for them is important. That's an area that's very understudied, in my opinion. Nutrition, if you think about things we do the most, we actually don't know that well how they work. Sleep, food, how that really reacts with you. And I would argue exercise and other things as well. So I think it'd be great to get a deeper dive on how all those things work, especially sleep is a good one. We know these impact your health great. So I guess where I'm going with this is that I do think we do need to measurement and find people's baseline. And we can find people. I'll tell you a fun story. There's one person in our cohort. They had a certain liver enzyme that was running at the low end of normal. And they suddenly saw it jump up. It doubled, but it still was in the normal range. So nobody said anything to that because our system, again, doesn't track longitudinal. But he came to me and said, Mike, my liver enzyme just jumped up by twofold. What do you think I should do about that? Nobody said anything. I said, well, go get another measurement. And sure enough, it went out of the normal range because it was headed in a bad trajectory. And we see this a lot. That's one simple example. And so, again, following your trajectory and seeing which kinds of things, and I think those will be run in the background in the future, what kinds of things shift you away from the healthy baseline. And the other, the optimizer, as you point out, how do you push yourself in good ways? Like, I'll tell you, if you wear a glucose monitor, I guarantee you'll change your eating habits. I don't know if you've ever worn one. People on this call have worn one. I highly, highly recommend it. I 100% will change your eat. You'll never eat the same because you'll see what spikes you. And then the logical thing to do is simply, eat the things that don't spike you and ignore those that do. But you got to know which ones do and which ones don't. And so the measurements are very powerful. And my company, January AI, they have a 32 million food database. They actually can make recommendations. Well, this food spikes you. So from 4 days of training, I think this is the future. We're going to have personalized AI programs built around people. And we can say, all right, this is what spikes you, this is what doesn't spike you. Eat this, don't eat that, and have this all built and personalized. And the goal will ultimately be, too, to change people's what their fiber and their probiotics to shift them so they'll become optimized as well. And then they may not have to, maybe they won't spike to anything if they can get their body properly optimized. I'm not sure. But that's the idea. So I think we'll get AI programs around ourselves. Not the language processing ones. I mean, that'll be used, but it'll be more the data-driven ones that will optimize your health. But I think that, and it is very personalized, and it'll require just collecting data and giving you feedback.
Absolutely. I continuously also track myself. I do not wear 3 watches, I could tell you, but I know.
I see the ring. I see you wear a ring.
Yes, I watched 2 and the glucometer, but I'm a not spiker. So I think for myself,
wow, you're amazing, a non-spiker. Yes.
Yeah. Vegetarian non-spiker and a good microbiome. But
nice.
What I learned, I think for everybody, knowing yourself helps you so much to adapt your health. But I think you would agree. But that's 100%. My last question before going to Dr. Walang Wang. So yesterday I was confronted with somebody who got the genotype, the epigenome, the microbiome, and the entire set of omics. And he said, okay, now I know who I am. And so what? Yes, I wore the glucometer for 7 days, and I see that I'm also not a spiker. So how do we then translate that into tangible, personalized interventions that we then, in the end, show, yes, we're changing the needle, and actually we have also a health benefit, and there was an economic benefit. So where do you think, how much can we really optimize while using all these devices and these measurements?
Right. No, great question. So obviously it sounds like this person's semi-optimized already, but he may or may not be as optimized as he thinks, right? So because it's still based on limited data. So one could argue, you could see, and there are people who take our yellow test, which is 500 analytes. It's that microsampling thing I told you about. We commercialize that. And there are people who are just green in all categories. That's great. But plenty of people will be off in one or two, and that's a little heads up to maybe optimize that. And it will require some level. Some of it's probably obvious what you can do based on data. You can say, well, your kidney things are up a little bit. You should drink more water. There's certain things you should be a little bit careful about. And certain medications can drive that, by the way. So, and this is where AI will also fit, pulling data and give you feedback. So I think this is going to be your most important health instrument in the future, possibly more important than your doctor. Doctors won't go away. They'll just use this sort of stuff, in my opinion.
I fully agree. Make us redundant.
I'm digressing a bit from your friend and but I think this will feed in. I'm so glad he hasn't found anything yet, but time, the number one factor for, all major diseases is aging and longevity. So the goal is, I hate to say it, but things will start popping up down the road, and he'll catch them early if he's following his trajectories. And I think that's where the world's going to be different in the future, really tracking people longitudinally, which you heard me say many times. I just think that's so important.
Absolutely agree. Thank you so much. Dr. Weilan Wang. Weilan is a postdoc in our lab. And Weilan, tell us what the audience is asking. And hopefully they ask what kind of variables Professor Snyder is wearing.
Yes, and thank you, Professor Snyder, for this very impressive talk. And we have a lot of questions coming up, so I will have to choose a few. So the first question from our audience is from our team member, Belinda Wang. She is our psychological expert. And she asked about how can we improve the compliance of wearable devices. She noticed that some of our participants describe stress or pressure while they're reviewing their outcome, and they feel like, oh my God, am I really have a spike, etc. And we also noticed that some people describe it as very inconvenient. So how can we improve the compliance from both aspects?
Yeah, okay. I think you did hit several aspects. So first, from the compliance, obviously you should find a device you're most comfortable with. Some people prefer rings, other people prefer watches. Both are pretty good, actually. And they're all pretty accurate for heart rate variability. Then they start differing when you get into other parameters. But so find the right device for you. I think in the future, quite frankly, there'll be implantables, and then you just pop it in you. And so that's where it'll go. We're not there yet. So I would argue, even with a little bit of inconvenience, it's still worth wearing these, because catching disease early is a big deal. So and so the other is on the manufacturer side. So the number one problem with collecting data is usually people keeping the devices charged. So the Fitbit and the Garmin's, they run for about 5 days each. The Apple Watch used to barely last a day, and then you'd have to charge it an hour and a half. Very few people would charge it for an hour and a half. So you want to wear it all the time. So it turns out, even though Apple Watch is higher resolution, it's not as good for most people because they let it burn out. Or they charge it overnight, which is even worse. It's one of the best times to do health monitoring. It's overnight. So bottom line is, getting the device is better, which is happening. The newer Apple Watch is better for battery life. Yeah, and I think that'll help with the compliance. Getting people to wear it. I think people wear it once you find something significant about your health. You'd be amazed, and I hate to say it, but COVID's been a bit of a wake-up call for people, that there is value to wearing these. So I think as you show utility and as they see their friends get utility with the devices, I hope that sucks them in. And to be honest, it's probably the best way to get people to wear a watch is to pay them. So I think you should get a discount on your health plan if you wear a device, because you'll actually probably walk your 10,000 steps. You'll catch disease earlier and such. So I think there could be financial motivations for making this happen as well.
Thank you so much. And this actually comes to another question from Jessica Lu, and she asked about can we select the most important features to measure from wearable devices? So we have a lot in market. So how can we choose the best combination of the measurements?
Yeah, great question. Number one measurement, I would argue, is glucose control, because the number of people who really have serious problems, I'm so glad Andrea has no issues or a friend, but I hate to say it, but in the US, 9% of people are diabetic, 33% are pre-diabetic. And I mentioned a lot of normal people aren't normal either, just don't know it. So, and those numbers are getting higher and higher. So imagine that right now, half the people have glucose dysregulation. So if we can catch that early, I'm quite confident we could we won't cure diabetes, but we might be able to help push it off for a few years, which should save the healthcare system a lot. So glucose monitor is a great one to have. When we talk about devices on watches, both resting heart rate variability, they're very, very valuable measurements. They're very sensitive. You can see when people are shifting off in their state. So heart rate variability, for those who don't know, it's a difference in timing between your heartbeats. It should be variable if you're healthy. If you're not healthy, it's actually consistently the same. So anyway, skin temperature, it's accurate on some, not on others, so on and so forth. And even when it's not accurate, though, the shifts are pretty good. So I think my own view is that if you're good at keeping your device charged, sure, go buy a high resolution watch. But if you're not, you better stay with a Fitbit or a Garmin, because they have a long battery life. And I think that's probably a better driving factor. Yeah, because missing data is the number one problem, I think, for this health monitoring. Back to your earlier point.
Okay, thank you so much. And how about this data? Most of the data are collected maybe in North Europe or in American. So does the data also comparable in other regions, such as in Asian and in other culture and diet patterns?
I'm sure the answer to that is yes. So you can track spatial heart rate variability is pretty easy to, and we definitely have these devices on a fairly diverse set of people. We have plenty of examples where Asians and specifically we can see COVID, resting heart rate jump up well in advance of COVID. So that's a pretty easy one. Now, it is true that there's one measurement called SpO2 blood oxygen that actually is a bit sensitive to skin color. And the darker the skin, the less accurate the measurement. But even there, it does work. It just doesn't work as well. And they're sampling enough. I think from our experience, you can actually still see shifts, because we have several African Americans in our study, and we are able to detect things. So that's the one that's the furthest off is the SpO2. It might be a little less accurate for other things, but in general, I think it's pretty powerful for heart rate and heart rate variability, which are two of the most important ones.
So we know that we can also use this in our countries too.
Oh, for sure.
Yeah. Thank you. That's a good news for us.
Yep.
I will be a more compliant user in the future after this talk. I will totally check my body temperature, SpO2, and heart rate.
Good. What are you wearing now, by the way? Are you wearing a smartwatch?
Yeah, I should, but you can say it's not here. But after this talk, I will definitely do that. More compliant.
Love it. All right, good. Yeah, it doesn't do any good at home.
Yeah, definitely. And another question from our audience, and he asked about, is there a better combination of when shall we start using these devices, and what is the best timing to get the optimal or the golden baseline of our health status?
Yeah, well, I would say, today or yesterday, start collecting data right away, because you need to know your baseline so you can see shifts. Now, an easy time of day to see health issues are first thing in the morning when you first wake up, where you haven't moved around too much yet. There's a lot of circadian patterns to biometric data. So, for example, your resting heart rate, your blood pressure, those are well known to change throughout the day. So, first of all, don't compare measurements at different times of day, because that won't be very accurate. But pulling your resting heart rate first thing in the morning is generally a pretty good way to see if something's off. Same with during your sleep. Sleep itself is a pretty good measurement for people's health state. So how well they sleep usually reflects, if the restlessness, which you can pick up with a smartwatch, that's all pretty important. Bottom line is, I guess, these are all pretty powerful. Did I answer your question? I kind of drifted off.
Yeah. I remember an old saying from China is that the best timing to plant a tree is 10 years ago, and then the second best timing is now. So maybe this is a good summary of that.
100% agree. I love that saying. Yeah, I better look that up. That's a great one.
Yeah, thank you. And I saw that our time is really going very quickly. So this is our last question. How far do you think is this multi-omics measurements to clinical practice in a more general public?
Multi-omics in general. So I think parts of what we're doing, again, what we do is a research study, but aspects of it, deep data will definitely move into the health system. So sequencing people's genome, I think, will become important down the road. I think other kinds of measurements, but RNA-seq, for example, wouldn't know what to do with an RNA-seq. Can't show that to a physician. So you need to distill out the relevant parts of that. You can still do RNA-seq, but say, look, here are the pathways, here are the shifts, like I showed you for the ageotype. So I think that we need to pull the information away, much like a pathologist distills an image into a final histology report. That's what we're going to do with the omics data. And mostly stick also with the ones that are most actionable. Metabolomics and metabolomics in particular is quite actionable. I think proteomics will be quite actionable as well. I think microbiome, we all know, will be actionable, but right now nobody's really has good interpretation or way to bring that in the clinical practice. It will in the future, though. Great question, though.
Thank you. Thank you so much.
And by the way, I'm happy to answer other questions. People can just write to me if they like. Okay.
Thank you for answering them.
We will forward you the questions. Thank you so much for this talk about really showcasing the next generation of medicine. It absolutely fits in the aspects, all the aspects we have in health and longevity medicine. What I learned is measure longitudinally. Of course, we already do that, but everybody should do it, and reacting on it and avoiding
reacting on it. Yeah, it's very, very key.
Okay. It was such a nice talk. I think that you as an audience also had a great time. It might have triggered lots of feedback, questions, other questions. You might also want to leave the comments. Please use the panelists and attendees option in the chat box. I also would like to announce, as in the last weeks, that we are very excited to open the registration of our conference, Unlock Healthy Longevity, using supplements from the 29th of February to the 1st of September in 2024. This is running here at the NUS in Singapore. And this conference is really shedding the light on supplements and bringing together leading scientists, clinicians, but also industry partners and the consumers and the regulators, which is very important. The next episode is on the 17th of August. The next speaker is Professor Lee from the Seoul National University. I will leave you, as always, with the video. And the only thing I can say, what do you regret? Take care.
What is your biggest regret?
I went to the playground and I want to go again today. Getting angry at my family sometimes.
Probably not trying as hard when I was younger with grades.
I regret taking myself too seriously when I was in high school.
Not getting to know my father more before he died.
My parents got me tickets for Christmas to see Sweeney Todd. I denied the tickets and my best friend went with another friend. I kind of regret that. I should have seen that musical with her.
See, I have many regrets, but I am where I am because of them, so I wouldn't change anything.
I tried to live without regrets. I think everything that I've done has been for a purpose.
Not staying single longer.
Not keeping in touch with people I probably should have kept in touch with.
Not listening to my self at times in life when I should have.
I can't say I have any regrets.
That I didn't get sober sooner.
I regret not joining the Peace Corps when I graduated from college.
That I didn't do all the things that I wanted to do, like go into acting much earlier.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.